Again, welcome all to this call that we do a version of each year around May 18th, and decided it was best not to do a call on a Saturday in recognition of HIV Vaccine Awareness Day. I'm going to provide a little bit of background, introduce two fantastic friends and colleagues to walk us through some of the scientific and advocacy priorities, and then most of all open it up for a conversation with them and with each other. Everyone should know we are in a muted mode to keep the line clear because we are recording the call and we'll make it available afterwards online. But we do want to encourage conversation throughout, and we'll give a number of instructions. If you're on the ReadyTalk interface, you can put questions in. If you want to email at any point to avac@avac.org, and then we'll also give some instructions on how to unmute your line later in the call to ask questions directly. As I said, May 18th each year is recognized as HIV Vaccine Awareness Day. It has a long and storied history. 22 years ago, when then-President Bill Clinton gave a commencement address at Morgan State University and used it as a chance to talk about the 21st century, which was soon to happen, to be a century of science, and he prioritized an HIV vaccine as a priority in his administration. And 2 years ago, on the 20th anniversary, we did a call with the leadership of the NIH's Vaccine Research Center, the VRC, which was really one of the most prominent outputs of that announcement in 1997 of an incredible investment in vaccine research. And we've come quite a long way in 22 years, but we still have quite a long way to go. Hopefully many of you will have seen that yesterday we released the toolkit for HIV Vaccine Awareness Day for 2019, and we present that kit, and we present this webinar and a follow-on webinar next week with a particular view in mind, which is one of remarkable progress scientifically, as you'll hear about on this call, in HIV vaccine research and development. And there's also been tremendous advances in other vaccine R&D activities in malaria, in TB, and I think many of you will know of the terrific advances around Ebola in a true emergency response to accelerate vaccine R&D. All of that happens, though, in 2019 in the midst of a very different narrative of vaccines, one that is where vaccines are questioned by some, where misinformation and myths are spread by too many, and we see a growing vaccine hesitancy. And actually next week's call will very much focus on that conversation because no matter how successful we are in developing a vaccine, if we can't deliver it, then a safe and effective vaccine actually doesn't do anything. And that really is this somewhat schizophrenic moment we are in vaccine science and advocacy today, one of great promise but of terrific challenge as well. And so we're delighted to welcome two people to give perspective on this, a terrific leading advocate and passionate community leader in HIV vaccines, Mark Hubbard, and also Mary Marovich, who directs the HIV vaccine research program at the NIH's Division of AIDS, and we're delighted to have them both here. Just before we start, I did with first Mark and then Mary, did want to just put one additional framework here, that as we talk about the advances in science and the challenges of vaccine delivery, this also happens at a remarkable time, and I think both Mark and Mary will touch on this a bit. But we sit here in mid-2019 with the 2020 targets that were ratified by the United Nations in a high-level meeting 3 years ago, the really beginning of the conversation of ending the epidemic. And you can see on the top of the screen here the targets for 2020, and we're giving the UN the benefit of the doubt that it goes till December 31st, 2020, not January 1st, 2020. But you can see that whether you mark it in the beginning of 2020 or the end of 2020, we are nowhere near on track to end the epidemic in the way that the mathematical models set out. And yet at the same time, that conversation about ending the epidemic, both internationally with the UN and more recently in this country with the current administration talking about ending the epidemic in the United States by 2030, comes at a time when we have many different modalities, as you see on the bottom of this screen, the vaginal ring, oral PrEP, long-acting antiretroviral injectables, and as you'll hear about today, antibody and vaccine research that is moving along with great excitement and with speed in a way that we've not seen before. And those trials will only report out well after the 2020 targets are missed. And yet we have these two different narratives, ending AIDS on the one hand and R&D on the other. So it's within that context that we really want to not only have this call and the conversation here and next week on the follow-on webinar, but really going forward together as advocates, researchers, policymakers, communities together, thinking about all of this together. And that's where we want to start today, and I'm going to first turn it over to Mark Hubbard, who has served for many, many years on the Community Advisory Board of the Nashville Clinical Trial Unit in the United States that has worked on vaccine and therapeutic research, and he also has been a very active leader in the Global Community Advisory Board of the HIV Vaccine Trials Network. So Mark, I'm going to turn it over to you to provide some introductory comments before we turn it over to Mary.
Sure, Mitchell. Can you hear me?
Yes, loudly and clearly.
Okay, great. So greetings from Nashville. We're not only the Music City, but this morning we're the capital of a pollen warning. So if my voice is a little scratchy, please forgive me. But I think Mitchell makes a good point. And I have to say, there's not anybody that's more excited about the progress we've made in HIV than I am. I'm eternally an optimist. I've been living with HIV for over 32 years, and I can honestly say there's never been a year that I didn't think that we'd made a huge amount of progress and that things weren't better than they were the year before. Still, the conversation needs to be more sober at times. Particularly, Mitchell and I were talking a couple of weeks ago. We were dealing with headlines about the partner study, and it's certainly exciting news that we get the most robust data we've ever had, that U equals U. But some of the headlines said things like end to AIDS in sight, huge leap towards eliminating HIV, end to AIDS is near. And, as advocates, we've got to kind of stop and get in touch with the very important realities that confront us. And so we have these innovative new prevention technologies that are extremely powerful, and they work very well in those that they work well in. So what I'm trying to say is they're helping us to reduce infections everywhere, and in some places and in some populations they've made a dramatic difference. But we also have to be honest and say, up until now, they haven't been able to help us in many of the people and many of the places where the need is the very highest. And so, as Mitchell alluded to, each of the new prevention technologies we have and each of the ones that are on the horizon have strengths and weaknesses. And I think all of them, to some extent, are faced with barriers that impede access. And those might be logistical, cultural, structural, political, psychosocial, things like stigma and medical mistrust, but those strengths and weaknesses are different. And what I believe is that a vaccine product could have a distinct advantage if it's reasonably effective and it's reasonably simple for users in terms of the number of vaccines they need over a given period of time. They might overcome some of these barriers in ways that other technologies that we've developed so far or that are on the immediate horizon do not. And I think there's some other reasons for optimism. If we zoom out to the big picture, and Mary's going to talk about this, we have sort of two major endeavors or tracks towards vaccine that are based on different but equally solid rational product development strategies. And I can honestly say, as an advocate, I believe they're being resourced and conducted and planned in a way that's pretty aggressive, and that's good news. And although we aren't at liberty to get into specific details on this call today, many of the folks that you know and love in the field, industry, government, advocates, organizations, are in the middle of collaborative work towards a phase 3 licensure trial that you probably will hear about soon, and it will be conducted in the most at-risk populations that are not being studied in the currently ongoing trials. So we can cross our fingers for that. If it happens, it will be happening, I think, significantly sooner than we expected. And although we can't read too much into that time frame, I think what we can be certain is that it does indicate a strong ongoing commitment to finding an effective HIV vaccine. And I'll leave it there.
Great. Mark, thank you so much. A fantastic introduction and a perfect segue into Mary Marovich to give us that scientific update. And Mary, I hope we can hear you now. If you want to be sure to hit *7 to unmute yourself.
Okay, can you hear me now?
Oh, absolutely we can. That's perfect. I'm delighted to welcome Mary Marovich, who, as I mentioned at the outset, directs the HIV vaccine program at the National Institute of Allergies and Infectious Diseases that's part of the NIH. And I think for anyone on the line who knows not just HIV vaccine research, but HIV research generally, that the NIH has been the driver of much of the research and a lead investor with many others throughout the entire decade's search for a vaccine. So I'm delighted to welcome Mary here. As I said at the outset, if people have questions as we go, feel free to put them in the chat feature or email avac@avac.org. And Mary, with that, I'll turn it over to you.
Okay, thank you, Mitchell. That was fantastic, and both you and Mark gave just a great framework for me to jump in here with the science. And I just wanted to say hello to everyone on the line, and thank you for joining. This is Mary. I'm calling from Washington, D.C. At the NIH. And I wanted to give an update and let you know how grateful and honored I am to participate in this webinar in honor of the HIV Vaccine Awareness Day. I'll just provide a little bit of historical perspective to kind of give you the background, and then hopefully convey our enthusiasm and optimism about the development of an HIV vaccine within our lifetime. Okay, so next slide, please. First, I'll just explain our mission for the development of preventive vaccine, and then, as I mentioned, I'll go over some key vaccine trials, and I'll explain, hopefully, the two-pronged strategy that the NIH is taking that includes both an experimental or empiric approach and then a rational or theoretical approach. I'll review the ongoing efficacy trials and let you know when the readouts from those would be available. And then I'll talk about the AMP study, the passive antibody efficacy trials, using broadly neutralizing antibodies, and then just close with addressing some of the vaccine development challenges. Okay, so if we have the next slide, here's just the vaccine research program mission, of course, to develop a safe and effective HIV vaccine. We have a fairly large and dedicated, talented team of scientists and administrators here at the NIH in the program that I direct, who support preclinical, translational, and clinical research. So we are a little unique in that we do from soup to nuts in this program. And the goal is obviously to put an end to the HIV epidemic. So we promote research supporting vaccine developers, academic investigators, laboratorians, clinicians who are working to discover and test novel vaccine candidates, and regardless of the funding environment. I think we all know the funding environment has gone up and down over the years. And basically, how do we do that? We rely on peer review and try to make scientific and data-based decisions. But we do also realize that partnerships are really important, and we've been focusing a lot on trying to bring others to the table, importantly companies and industry to support us with their know-how and ability to produce vaccines. So next slide. We can just skip that slide, the preventative vaccines, and let's just go right to the first science-y slide, and that's the RV144 graph that many of you have probably seen. This is sometimes referred to as the TIE trial. It was the first vaccine to show prevention or protection from infection, and it really rejuvenated the field. If you all remember, those results were published in late 2009. I can't imagine that it's a decade since that's happened. But it was important because it was the only vaccine to ever show a reduction in risk of acquisition. And if you look at the two curves, you can just basically see on that Kaplan-Meier curve on the left-hand side is that those lines separate. And you can see that on the y-axis, it's the increased risk of infection or the percentage of infection, and on the x-axis is time. And you can see that the vaccine arm in green is lower than the placebo arm, and that persisted over time. The study actually was fairly long in that it was carried out for 3 years after the final vaccination. And if you look at the graph in the lower right-hand curve, you can see the very top row there at the 12-month time point, and what I have circled on the graph, it actually shows a 61% reduction in vaccine efficacy of 61%. And that, however, waned over time. If you look at the 24-month period and the 36-month period, it drifts down towards 31%. And this, as I mentioned, created a lot of excitement. You'll remember that. And so what we thought was really important, and this is one thing that NIH focuses on a lot and devotes a lot of resources to, and I'll explain why, is trying to identify how these vaccine works. And that's called usually a correlates of analysis or a correlates of protection. And we usually try to find what is the immune system doing, after vaccination that has conferred protection. What kind of immune responses, antibodies, T cells. And interestingly, it is in this correlate study, it was recognized that non-neutralizing antibodies correlated the presence of those antibodies, specifically directed at the V2 area of the envelope, were associated with a reduced risk of HIV infection. And I'll get back to these correlates of protection about how important they are, because if we know what is the right response we want to generate, we can often do studies quicker by what we call bridging to immune correlates, and we'll get to that in some further slides. So as a result of the RV144 trial, we formed a partnership, a large partnership called the P5 program, many of you have probably heard of, and that's the Pox Protein Public-Private Partnership, and now you know why we call it P5. And it was a group of partners, including Sanofi, who made the Alvac pox vaccine, and it was Novartis, then GSK, as far as the companies, the Bill and Melinda Gates Foundation, the NIH, the Military HIV Research Program, the Medical Research Council in South Africa, and of course the HVTN to implement. And the focus there was to basically substantiate and improve upon the RV144 result, because if you get a good result in science, you always want to make sure that it's real, and there indeed was a lot of skepticism about this result. So the P5 developed vaccines that were relevant for South Africa, a place that is heavily burdened with HIV infections, and they were directed towards the circulating viruses, clade C. And we decided to make a strong effort to try and verify that result and improve upon it by using a different adjuvant and increasing the numbers of vaccinations. So if you move to the next slide, I can explain the NIH's vaccine strategies. It's what we call a two-pronged strategy, and it may look a little busy, but I'll break it down. If you look on the left, in the upper left, the RV144 TIE trial, that's one of the prongs of approaches called empirical. So we're just basing our evidence on those empirical results from the RV144 trial, saying we got these immune correlates. If you move to the right, let's develop a vaccine to improve the potency and durability by directing it towards the immune correlate that was identified. And then if you move to the further right, you can see in green there's two efficacy trials that are sort of modeled after that non-neutralizing antibody protection. So it's HVTN-702, which is a phase 2B using the Alvac clade C GP120 with an MF59 adjuvant. That's the so-called P5 program efficacy trial. And then below that, HVTN-705 is a phase 2B using another viral vector called Ad26 with mosaic inserts and using GP140 envelope boost. So again, viral vector primes, envelope boosts The first is the P5-702, and then the subsequent one is what we refer to as the Janssen collaboration, and I'll speak about that. And then back to the other prong of the approach is the theoretical, again on the left-hand side, the broadly neutralizing antibodies, because we know from animal models that if you give passively primates some broadly neutralizing antibodies, actually isolated from humans who've been infected with HIV, if you infuse those, you can prevent infection in a dose-dependent way in the animal model. And that's very, very strong evidence for a correlate that if you have these antibodies around, it should prevent infection. So our other approach is to try to develop vaccines that would induce these broadly neutralizing antibodies. We know from people who are infected with HIV that they can develop these antibodies, so we know the human repertoire can make them. But we just have to figure out how to induce them. And then the other approach within that broadly neutralizing antibody strategy is we've been trying to understand if that would really work. So we've taken a broadly neutralizing antibody and we're doing an efficacy trial, it's called AMP, antibody-mediated protection, to understand, to prove the concept, do these broadly neutralizing antibodies actually prevent infection? And those studies are HV10, 703, and 704. Those are the AMP trials, and I'll talk a little bit about those. Okay, next slide. All right, so here we go. We can go through this a little quicker now that I gave you that background. So the 702 study, as I mentioned, is the P5 study. There's the schema. You can see there are many vaccinations. It's a study of 5,400 participants. I believe it's almost completely enrolled. It should be enrolled this month. It's a 1-to-1 randomization. Half the participants get the vaccines and half the participants get placebo. The study officially will read out in 2022, but we should get interim results in 2021. And again, that's the follow-up to RV144 happening in South Africa. Okay, can we go to the next slide? Now, this is the other efficacy prime boost vaccine study that's aiming for these non-neutralizing antibodies that we saw in RV144, and that's the Janssen collaboration. And the interesting thing here is Janssen is interested in developing a global vaccine, what they call global coverage, by using a mosaic insert within this viral vector. And that basically stitches together genetic information that would induce protection from theoretically all strains of HIV, as you can see. So they have the strains on the right-hand side that they're aiming to protect so that we could really just use one vaccine instead of targeting it towards different regions, which has been more of a strategy for the P5 program. So again, it's viral vectors that have these mosaic inserts, and then they're boosted by envelope vaccines. If you can get to the next slide, I think I can show the schema there. Yeah, this is 705, and this is a study, again, that's almost completely enrolled. We hope that it'll be enrolled at the end of this month. It's being conducted in sub-Saharan Africa, and again, it's a 1-to-1 randomization. It's a smaller study, so it's more of a proof of concept efficacy study, but still we're looking for an efficacy signal. And the interesting thing about this 705 study was it was really pivoted off protection in primates. So we don't have a correlate for this, but the primate data was so compelling and the company was so interested in testing out this global vaccine strategy that they've invested an enormous amount. And as Mark has alluded to, the company is planning a phase 3 study that could be registrational. And so their investment is really key to the field, that without a protective signal, they're going beyond this small 2B proof of concept efficacy trial and moving right into a phase 3 that, as Mark said, we think will start for sure this year and maybe sooner than we think. So that's very exciting. Can you get to the next slide? Okay, so anyway, I've been talking a lot about these non-neutralizing antibodies, and I just wanted to, for those of you who aren't really familiar and don't do this every day of your life, I'd like to just review the structure of an antibody and what we're talking about non-neutralizing and neutralizing. So Antibodies have been around, obviously, for a long time. They're a great form of protection, and they're not just a one-trick pony. They have many ways of offering protection, but we often just talk about neutralization, which we know is a correlate of protection for a lot of vaccines. So basically, if you look at this Y-shaped molecule on the left, the upper two, say, rabbit ears, that's where the antibody really latches on to the virus and actually can cover the virus and prevent it from entering the cell. So that's thought to be neutralization. And that concept of using a neutralizing antibody against HIV is being tested in the antibody-mediated protection, the VRCO1 study there, as you can see on the right-hand side. Whereas the non-neutralizing antibody approaches, the 702 and 705 on the right-hand side in the blue box, they're actually looking more at binding. So they're non-neutralizing, but they do bind to the virus. But they use the other end of the antibody molecule, what we call sort of the non-business end, which is an Fc receptor that can bind to other cells and can help to kill infected HIV cells. So that's so-called Fc-mediated, which is non-neutralizing. And what we're hoping is that in the end, perhaps we could have an additive efficacy effect where we could induce both neutralizing antibodies and neutralizing antibodies that could kill infected cells via non-neutralizing mechanisms like we saw in RV144. Okay, so next slide. I'll move into the broadly neutralizing antibody area, and this is my last topic, really. So what about broadly neutralizing antibodies? It's kind of a big buzz. I'm sure you all have heard of at least the AMP study. And it is on the basis of these broadly neutralizing antibodies that people who are infected with HIV make. And so it's really dramatic. We didn't understand that people who were infected with HIV make broadly neutralizing antibodies. The problem is it's just sort of too little too late. The virus is always outrunning these antibodies because the virus mutates so quickly. So it's not effective in people who are infected because they're already infected. But the thinking is if we could get these antibodies on board before the virus comes in, it could work to prevent infection. So the concept is that we know the human immune system can make them, but we need to figure out if we can get a vaccine to make them. There are many viral targets, at least 5 now, vulnerable sites on the HIV virus that these broadly neutralizing antibodies target. They're sort of unique antibodies in the sense that they develop really slowly. In adults, it takes several years, and they may develop quicker in infants just due to the differences in the maturity of their immune system. It's thought that they develop like on the order of a year, whereas it takes several to many years in adults. They're highly specialized antibodies. They require a lot of mutation and refining and developing all these long loops. So that's why it takes so long to develop them. But we know, as I mentioned, in the non-human primate challenge model, they offer protection in a dose-dependent and repeatable way. It's very convincing. What we don't know is if they'll protect humans, right? That's why we're currently testing that in AMP. And then the other thing we don't know is can we find vaccines that will elicit these broadly neutralizing antibodies in people? The one thing we are pretty sure about is that we're making progress in that latter bullet about vaccines potentially eliciting those, but we may need a multi-component vaccine, and I'll talk about that in the next slides. If you could advance, that'd be great. So again, I talked about most effective vaccines induce neutralizing antibodies against the virus, and these are some pictures, very high-resolution pictures of viruses, hepatitis B on the left, influenza in the middle, and HIV on the right. And there are antibodies, of course, that develop to all these little proteins on the surface of the virion, and those surface molecules are often used to bind to receptors to get infect cells. So one thing is you may notice by looking at these virions that there's not as many as those proteins or envelope spikes on the surface of HIV, and that's one of the immune mechanisms we think is that's why it's harder for the immune system to find them because they're so few and far between, and the antibodies can't cross-link between each of those spikes, whereas they could easily cross-link, as you can imagine, in the hepatitis and influenza models. So, okay, we can go to the next slide. So anyway, as I mentioned, it is on the backs of those folks who have been infected, to be perfectly honest with you, from natural infection, that we've learned and that they've contributed so greatly to the vaccine field, is that the human immune response can produce these broadly neutralizing antibodies. And in fact, they've been isolated directly from people who have been infected. And I just wanted to show you this cartoon on the right that has multiple sites where the broadly neutralizing antibodies target. We can go to the next slide. And the last big study I wanted to talk about is, of course, the AMP study, and that's a phase 2b study to evaluate the efficacy of a particular broadly neutralizing antibody called VRCO1 to see if it would reduce acquisition to HIV infection. There are two studies that are very similar, almost identical, except that one study is being conducted in the Americas in men, and in the other study is being conducted in Africa in women. Okay, so we can move to the next slide. Oops. I just lost my, hello? Can you hear me?
Oh, we can hear you, Mary.
You can? Okay, sorry. I just lost my, my computer screen, but I'll just work off my slides here, if that's okay.
Oh, no problem. Right now it has the monoclonal antibody up.
Okay, good. So basically, this is just a picture for those of you who are interested of what the VRCO1 monoclonal antibody looks like. As I mentioned, it was isolated from an individual who was HIV infected for a long time who maintained virologic control without taking ARTs. It was isolated and developed further by John Mascola and colleagues here at the NIH. And the important thing to remember is this broadly neutralizing antibody blocks the CD4 binding site. And you all know that that's a critical part on the CD4 cell where the virus enters. So since binding to CD4 is a key process to infect the CD4 cells, this is a broadly reactive antibody. It can protect against many strains because that's a common theme in order for any virus to get in. And as I mentioned, it's being tested in an important set of trials. If you go to the next slide, we can look at the AMP study schema. And I'm sure it's coming up there. So this the AMP study is designed very carefully. It actually has three arms in this study, or three groups. Two of the three groups receive the VRCO1 antibody via IV infusion every 8 weeks, and they either get a 10 milligram per kilogram dose or a 30 milligram, so a lower dose and a higher dose. And they've given every 8 weeks for 10 doses, and then their HIV infection rates are monitored. The study is, as I mentioned, in two different locations, as shown on the slide, in high-risk women in Africa and in high-risk MSM and transgender individuals in North and South America. And the study has completed enrollment last year in September and October, respectively. And we expect results of this study next year. So it's really exciting. And again, the important point is this is really a proof of concept. It's not a proof of product because we're not sure that only one broadly neutralizing antibody is enough. But if we do show some protection and some selection of viruses that are, prevented from infecting people, it'll be a very important proof that broadly neutralizing antibodies can protect. So the last, if you want to go to the next slide, this is the last really heavy-duty science slide on a vaccine that I want to talk about. I'm presuming that you have the fusion peptide vaccination with trimer boost slide in front of you now. And the only reason I show this slide is it's a little busy, but the important point is if you look at the upper left, there are these a series of vaccine products that are being given. So there's this fusion peptide, there's 2 of them, and then it's followed by these 3 trimers. And this study was done in all different animals, small animals up to non-human primates at the Vaccine Research Center. And they took this fusion peptide that is common to all HIV virions. It's near the base of the membrane. And again, they found that antibodies against this fusion peptide can cross-neutralize almost 60% of the viruses in a 208 viral panel. And we've never really been able to see that. So I just want to draw your attention that this is a very exciting milestone that we now can induce through vaccination antibodies that are cross-reactive to, in some animals, 59%, preventing 59% of those viral strains. So this means that this strategy is promising, and we'd like to test that. Those products will be tested this year in humans in phase 1. So if you go to the next slide, this is just, there's way too many vaccines to enumerate in here, but I wanted to let you know that we are working collaboratively, the CHAVI programs, the NIH, and other investigators in Europe, Robin Shattuck, et cetera, Rohir Sanders, and many others are developing these types of immunogens that could theoretically induce broadly neutralizing antibodies. And again, I mentioned there will have to be some type of sequential vaccination, and if you move to the next slide, it's called the virus neutralizing antibody coevolution slide. This is a kind of an old paper, but I wanted to just reinforce the idea that there's this evolution of neutralizing antibodies that happens in a certain percentage, maybe a fifth or a quarter of people who are HIV infected. So if you look at the arms race, underneath that there's the initial virus that infects a person, the transmitter founder virus. That will elicit an antibody. So the immune system will make an antibody, but by the time that antibody forms, the virus has already mutated and it's resistant. So if you can see that sort of zigzagging across there, by the time the antibody forms, the virus is mutated again. And so what happens is this antibody repertoire just gets more and more refined, and it becomes better and better to neutralize a large variety of viruses. And that's how we develop broadly neutralizing antibodies in humans. So we think that our vaccine strategy might have to mimic that, in that we would have to have multiple components to our vaccine, and probably in a sequential fashion. So if you go to the next slide, we're coming down the stretch here, folks. This is a graphic that I make reference to, the sequential HIV immunization strategy. And the reason I show this again, this was taken from Dennis Burton's review in Immunology early this year in 2019. And basically this is just a graphic or a cartoon that shows how the broadly neutralizing antibodies have been isolated from humans, and how we use those as sort of a template, you know, to instruct how do we make the vaccines from the business end, that Y portion that I talked about that binds to the virus. So we use that to break down which parts of the virion, what types of envelope immunogens would be able to stimulate these broadly neutralizing antibodies, and we define the sequence of immunogens that would be required. Then we test them in small animal models, moving up into the primates, and if it looks promising, we then move them into humans, into clinical trials. And that's where we are today. So I'd like to just close with the next slide, where I summarize the efficacy studies and provided timelines for you. I presume you're on that slide, or will be soon. So basically, you'll remember we have the P5 study, the LVAC GP120 MS59, which is an RV144 follow-on study. That's 702, conducted in South Africa, men and women, 5,400 N, should be enrolled this month. And we will have final results in June 22, but interim results probably in 2021. The same thing for the AdMosaic 705 study, conducted in sub-Saharan women, an N of 2,600. The results should be again in June of 21, at least interim results, and final results in 2022. Next, we have the AMP studies. As I mentioned, those are conducted in 2 different geographic areas, in sub-Saharan Africa, America, and Lausanne, I should say, in Europe, in women and men. And those results should be, interim results should be happening next year, at any time next year, the latest September. The final thing I wanted to say is that I'd like to encourage you to watch for these early phase studies that are coming out that are pretty exciting regarding these molecules that can induce or at least start a neutralizing antibody line in humans, like the fusion peptide that I referred to. So keep an eye on those phase 1 studies. They're getting exciting now with the ability to elicit neutralizing antibodies. And then finally, I'd like to just close with this graphic on addressing HIV vaccine development challenges. This is just a graphic that we like to refer to, which tries to bring in everybody who's involved in the whole HIV vaccine enterprise. And it's a slow, it's a slog. It's not sexy developing vaccines, but I think if we're persistent and we work together, we can get there. So if you look at the part of the circle where there's a syringe, we talk about identifying immune correlates in any protective vaccine studies. So that means even in primate studies, our best animal model, if we see a vaccine that induces protection from challenge, we try to drill down as far as we can to understand what those immune responses are so that we could one day maybe say, oh, we know if you develop this immune response, it should be protective. And that could be really informative, particularly in the era now where there's a lot of other prevention modalities. We're probably not going to be able to do multiple very large efficacy trials as we've been doing in the past. So we use these correlates, if you go counterclockwise, to advance vaccine candidates and hopefully incentivize industry to join us and de-risk them. And that would increase capacity and efficiency of vaccine manufacturing. It would help us pre-qualify vaccines for potential registration and marketing as we move towards the 9 o'clock part of the circle there. And what companies can do is they can develop platform technology where they use a similar backbone for say, Ebola or Zika, and that type of thing is even happening. So finally, at the center of this all is the people part of it, in the sense that we need public-private partnerships to work together to get this done. And in red, the red circle is really the community that sort of envelops the people coming together, because we know that we can't do this without the support of the community and the involvement in the community. It just wouldn't be able to be done, and for that we are very grateful. So we can move to the acknowledgement slides. And just again, I'd like to thank everyone who's involved. And today I'd like to really specially highlight vaccine trial volunteers, those who are interested in supporting us, and our community partners who've just been there all the way. We hope to really deliver. So thank you, and I can take questions.
Great. Mary, thank you so very much. That was terrific, and a lot of information there, but really beautifully delivered. There are some terrific questions that come in, and I'm going to get to this in just a second. We do have limited time, but I know maybe I'll start with you, Mark, if you want to just at any points there, as Mary ended at the important partnerships and clearly the role of community in all forms. And I wonder if you want to add something very quickly, and then we'll open it up to questions.
Sure. I'll just make a few points if that's okay, Mitchell. And I love that last graphic, because it's absolutely true that we not only is community critical to getting this done, but that as we strategize and plan, we have to keep community involved. And I would say sort of the challenges that we're facing from a community standpoint and the conversations we continue to have are not new, but they're intensified for sort of two major reasons. One is that we're doing this work in the context of ever-increasing PrEP use. And the second is, and this is a good news, bad news situation, that we can expect from these new products to see more consistently and more successful products soliciting a more robust immune response. I mean, that's the goal. So in the first category, for example, it's always been hard to reconcile this necessity to recruit the people who are at high risk, that are truly at high risk, while at the same time making sure that they have access to the best tools and have the potential to succeed with those tools. And PrEP makes that harder and harder. The more we move forward. And it makes it a little bit more hard to explain how we do that in an ethical way that is respectful to the community. In other words, we have to design and conduct trials in a way that protects individuals from unnecessary risk and, in fact, helps them to reduce their risk as much as possible. But on the other hand, we want to protect the community as a whole in that we don't want to use resources to do a trial that can't answer the question of whether the product works or not. And so these are difficult and subtle matters. We've been talking about them for a while, and we're pretty good at talking to people who are potential trial participants sort of one-on-one or after they enroll in the trial. But I think, you know, as AVAC would say, good participatory practice, or GPP says, we must also be able to have that conversation with the general community that surrounds the people who participate in our trials and who may benefit from the results of these trials. And that's very important, and I think it's a little bit more challenging because they may not have the research literacy that the folks on this call sort of take for granted. And finally, on that immune response point, we really think at this point that the trials we're conducting now, these large trials, are going to present the reality that people who participate in them are much more likely to experience what we call VIS, vaccine-induced seropositivity. And that means we think in these newer trials that more people are going to need special testing to find out their true HIV status, and they may need that for a very long time. So again, VISP is not new, but it's becoming a more intense challenge. And so in a broader sense, we have to ask, what are the implications of living in a future in which a lot of people, a lot of participants, and more persons in the general population have this? And we need to think about that. And, you know, if we're successful, what does it look like where we have a lot of vaccinated folks walking around who have these antibody responses, and what kind of technology do we need to help them determine if they have acquired HIV? And I'll leave it with that.
Great, Mark. Thank you so much, because I think what you've just added to Mary's fantastic presentation is, you know, the reality of trials designed and conducted in 2019 and going forward are in a very exciting but complex world. So thank you for that. And it actually relates to some of the questions that have come in. I'm going to try to ask a few real quickly, and we'll see how far we get. But we had a great question, Mary, that was triggered by something you had said early on in talking about the current set of efficacy trials and talking about the 2B trial in South Africa of the Janssen Mosaic, and then the proposed phase 3 trial, and you described it as a potential licensure study. So a very careful listener, advocate, asked, what's the difference, and why is one licensure and one is not? And are those two studies, the one that is in South Africa with Janssen and the one that's being planned, is this all working towards the same vaccine to be licensed? If you could help people understand that.
Yes. Can you hear me?
Yes.
Okay, thank you. Thank you, Mitchell and Mark. That is a great question, and I'm sorry I wasn't clearer about that, but it is somewhat, you might get somewhat variable definitions of what a registrational trial is. Some people will call trials pivotal trials. There are certain regulatory definitions, but the 2B Janssen trial being conducted in South Africa is a smallish study. It's, I think, like 2,700 participants, and that's relatively small, say, for an HIV vaccine trial. You know, typically they're in the 5,000 to 6,000 range. So it's a smallish study. It is powered to give us a good answer, but it's would be the first. If there was a positive signal, it would be sort of the first pillar and part of a package that could be submitted to the regulatory authorities for registration and approval to be marketed. But you usually would require more than one smallish study for the regulatory authorities to allow you to advance that vaccine to market. So it's not uncommon for there to be more, multiple studies, the results from which would become part of a package that's submitted for registration and approval. And the other point is that's important about these two studies that Janssen is conducting is that it's mostly a clade C epidemic in southern Africa. So the 2B is directed, is testing that concept. Can this quote unquote global mosaic vaccine prevent infection from clade C? And then the phase 3 study, which would be conducted in other parts of the world with presumably other clades, that would further that concept to say, okay, maybe we can protect clade C. Is this really a global vaccine? Will it protect in other parts of the world? And that study is conducted will be conducted in more areas, presumably, and is somewhat larger, and will be submitted if that's supportive. Both studies would be part of a package to support advancing and registering those vaccines. The other point that I didn't make really clear, and this is another great pickup by the person who raised this question, is there is a slight difference between the vaccine regimens. So a registrational trial for sure is going to have the final vaccine regimen, right? You're going to want to test exactly what you would be proposing to bring to market. The 2B trial being conducted in South Africa has only one envelope protein boost. The planned phase 3 study has 2 envelope proteins. One was subsequently developed, and so there's actually like 4 components to the vaccine. There's 2 adeno vectors, and then there's 2 proteins, and those are contained in this planned phase 3 registrational trial. So it's a little bit of many different things, but the 2B trial being smaller doesn't have all 4 parts to it. It has the clade C envelope protein that's relevant for Africa, but the larger study that could be a potential registrational quote unquote study would have this mosaic protein that then could provide coverage further to other parts. Does that help at all?
That helped tremendously, Mary, and it wasn't that you didn't cover or confuse. It is complex as vaccines get developed, and that was a really terrific explanation. I will just say on the screen you will see, and actually the person who asked the question, a wonderful advocate in Europe, just said that was super helpful on the chat box. But just to say on the screen, for those who are able to see the screen still, you'll see just some thumbnail of a series of graphics that we released yesterday for Vaccine Awareness Day. There is one more that is in development that looks at this, with the adeno 26 vaccine. That's the backbone of what Mary's describing, coming from Janssen in partnership with many. And we're developing an infographic that looks at each of the trials that have contributed to first triggering the phase 2B trial in South Africa, or Southern Africa I should say, and soon this larger phase 3 study, and try to show what each of those trials was contributing towards in that process, which actually relates to another question that came early on, because you reminded us at the very outset, Mary, that we're almost 10 years since the RV144 results. I can remember it all too well in September 2009. And someone asked a question, are there lessons that you, Mary, and you, NIAID, and the field have learned over the decade to ensure that if there is a positive result from any of the trials you described on your presentation today, that the field can move faster, that we aren't waiting another decade after a result that was promising to be where we are today? So are there key lessons that you take away from the last decade from RV144?
Oh, definitely there are, and I believe that there was actually a paper written about this, because we felt very strongly about communicating lessons learned, and I'll send that reference to you, Mitchell, and perhaps you can make that available or a link somewhere. But one of the main things that was important after RV144 was that, to be honest, I think people were a little bit surprised that there was protection, and it became apparent that, hey, if there really is a vaccine that's modestly, maybe more protective, we need to be in a position to distribute that product. We need to be in a position to just kind of flip the switch at the manufacturing facilities and make sure we can get the vaccine available to people in a very rapid fashion. And what happened with the one of the components of the vaccine, the AIDS vax, there was a sort of uncertain future for the company and the production of that envelope vaccine. It was a bivalent, a combination of two proteins. So part of the scale-up and manufacturing and rollout and demand forecasting and all those types of things we realized were really important, because you never know if you're going to get an actual good response that you'd have to take action on. So as a result of RV144, we developed a roadmap working group, and there was a number of people that are involved in that, and there's many more planning sessions, stakeholder involvement, just scale up and manufacturing plans. We've learned a lot about what we would do post-positive signals. And I think that's a great question, but there are a lot of lessons learned.
Great. Mary, thank you. I know we're near the top of the hour, but people are still on, and we have some great questions. So I'm going to keep going and hope that Mark and Mary can stay. Next week at the same time, and we'll put up the details in a minute, we're doing a call focused on kind of current conversations about vaccine hesitancy. We're certainly seeing it with a rebound in measles, unlike what we would have ever imagined. But a question that came to you, Mark, as someone as was listening. Do you see, as you talked about community engagement and recruitment and good participatory practice, do you see the current movement around vaccine hesitancy for other vaccines? Do you think it's going to, are you seeing it yet, or do you think it might affect recruitment in the trials, as Mary was describing, this exciting trial landscape going forward? What do you think is going to play out, and have you seen any of that already in your work?
I think that's a great question. I think it's very hard to predict. What I would say is, to the extent that it exists in the field of HIV, it would not be new, just as it's not new in these other fields. So in fact, what we're seeing is long-term repercussions of sort of having this thing simmering on the back burner, and the community seeing that it has very real impact. I think we're at a different stage. So we don't have a vaccine we're trying to deliver. But I think as we think about how critical these conversations are, and we learned some of these lessons with PrEP long ago, as you well know. If we don't have robust understanding of what we're doing in terms of who we say is eligible to participate in a trial, who is not eligible to participate in trial, and gosh, this is a huge issue. If we don't make sure that the community at large understands that people who have vaccine-induced seropositivity are not in fact infected, then we are going to face very similar problems. And so I think, again, being able to deliver these messages on all levels, okay, not just, sort of the research geek level or the advocacy level, but to the persons who live in these communities is very important. And I think it's a cautionary tale what we're hearing with, just dramatic reports of how these things have been manipulated by people who seek power in terms of the Ebola outbreak is something that we just have to be 100% prepared for. The conversations at all levels, as GPP dictates, not just community, but institutions and authorities and governments, law enforcement, all across the spectrum, just haven't ever been more critical. And I think if you do them in advance, the work is, an ounce of prevention. I'll just leave it at that.
That is terrific, Mark. Thank you. A great answer. Again, people are still on, and there were a couple of questions that I think are really important about some of the antibody work. And Mary, you described that the VRCO1 is not a product development trial, but rather a proof of concept. And actually, on the screen, you mentioned some of the combination antibodies. And one of the new infographics, thanks to great input from a number of researchers, we developed this graphic at the bottom left about some of the combination antibodies that are moving into trials, much not unlike how we did antiretroviral research years ago, combining ARVs. But first, a question about the antibodies, because someone highlighted here that when you talked about the antibody that was isolated that became VRCO1 was from an individual who was infected but who had, I think you said, who had virologic control without antiretroviral therapy. But the antibodies alone for someone who's already infected don't control the virus without ART normally. So can you just help clarify what that interplay is? And I know you had a great slide on that, which I can go back to, but just wonder if you could talk a bit about the antibodies that are working or developed in an individual who's already infected, and how that all plays out and what the implications are.
Yeah, it's a great point, and I realize that that's sort of seems a little contradicting. For some reason, the individual who was being followed here at the NIH was able, maybe through some other mechanisms potentially, able to control, or maybe genetically, they were predisposed to being able to control their virus in the absence of ART. Now, this was quite some time ago as well, so back in those days, people probably didn't start ART until later course, unfortunately, in their infection. But it's an odd thing. In fact, in many of the people who develop broadly neutralizing antibodies, they actually tend to have pretty high viral loads, and they think that thank you, Mitchell. This is the slide. If you think about it, unfortunately, it's thought that this high level of presence of the virus that's continually mutating somehow educates the immune system, the antibodies, to develop into a really strong neutralizing antibody. So it's kind of the yin and the yang, in the sense that the virus it continues to escape, but it eventually educates the immune system on how to contain it. But it's never enough really for an individual to clear their infection, largely because that particular broadly neutralizing antibody, their virus has escaped it all along. So it might not work for them, but it may work for other people. And maybe you're trying to think, so this VRC isolate is a little unusual in that it did come from a participant, from a individual who was infected that did contain their viral load. But most often we see it in people who have a elevated viral load that can really drive this evolution of the broadly neutralizing antibodies. But it's one important point is we do think that combinations, as Mitchell has mentioned, combinations of these broadly neutralizing antibodies, if multiple of them given at the same time, much like an ARV, they could be potentially used instead of drugs. So you could theoretically maybe give someone a cocktail of, say, 3 different broadly neutralizing antibodies with a long half-life, and potentially maybe they could just take that once, for 6 months and then stay off their drugs. And if they could keep their viral load down with multiple different broadly antibodies that could, prevent the escape. So I think it's this idea that you need to still be very aggressive in the setting of ongoing infection to nail down every escape hatch for the virus. It might be easier. And we might be able to use fewer antibodies to prevent infection just because there's fewer virions around and less mutation. But for someone who's already infected, we actually think you might need several. So hope that
that's super helpful. And Mary, just to pick up on that, so it sounds, from what you're describing, there is a pretty clear analogy to treatment. So, we think about triple combination antiretroviral therapy for treatment, but in PrEP, we have 2 drugs, and one study even showed tenofovir alone as being effective PrEP. So you're describing something similar. 1 or 2 antibodies may be in prevention, but a triple combination of antibodies may be future therapy as well.
Perfect. Yes, exactly. Thank you.
Great. No, super, super helpful. And I will say, just thinking back on the 22 years of vaccine awareness days, the kinds of things both you and Mark are talking about were inconceivable in May 1997 when Bill Clinton gave that commencement address, probably thinking, like many of us did, that we would be much faster to an HIV vaccine than we are in 2019. But yet I think you both described why this is a moment of remarkable both excitement but also remarkable complexity going forward. I know we're past time, but maybe on that, would love just maybe to offer each of you a chance to give a perspective as you think ahead to both what is perhaps one of the largest challenges you are seeing and one of the most important opportunities. And feel free to combine that into a single thing. But love to have you both just really briefly say, what are you seeing right now sitting here in May 19 as both biggest opportunity and biggest challenge. Who would like to start?
Please go ahead, Mark.
Oh, wow. Okay. So one of the things, and I will say that Mitchell knows this is a soapbox that I get on, I think we need to guard against advocacy fatigue. And that's not just for the advocacy, the people we call advocates, for the community at large. And I think we've done a really great job of creating excitement and maintaining excitement for this research. Some things produce more dramatic results more quickly, and so they tend to catch people's eyes. But the reality is, in general, research is a step-by-step process. So we need to always be having the conversation and always be aware that people want to know what's new and what's different. And then on the human side, I think it's really important that we take a look around and see who's involved and look at the demographics. I think that we can always use fresh blood and new perspective, and I would like to see a lot more people who are way younger than me who are ready to move up that sort of ladder, that sort of advocacy ladder from sort of being excited about and talking about things at the ground level to actually getting involved in committees and boards and groups that are looking at the hard questions, and then some of them moving up to really hopefully becoming interested or becoming science geeks and doing that work as well, because we need people involved at all of those levels and for them to be talking to each other, and not just about age, but race, ethnicity, resource limited setting versus resource rich settings, people of different genders and people of different orientations, to be actively involved so that we can do the best work we can do. And so I'm looking forward to seeing people who have not been as well represented at the table come join us as they are already doing.
Great. Beautifully said, Mark. Mary?
Oh my goodness, that was so great, Mark. I second that. And I guess the challenge I would see ahead of us are, just like we've been saying here, is that vaccine development is a slow process. It's a longer-term solution. We hope that it's something that could happen within our lifetime, and it should be accessible to everyone. We would make sure that the Gates Foundations and others have those efforts to make sure things are available to people. And I think it's important to keep people safe in the meantime. I think we need, as we do, to continue risk reduction counseling and recruit those participants who are interested in developing a vaccine. and just we understand that this is slow, but don't give up on us, because I think that it's becoming a very exciting time, and we'll work to make sure that there's resources and that HIV remains a relevant concern for our research.
Great. Mary and Mark, both of you, thank you so very much. And I think for me, one of the most exciting pieces to hearing you both speak is that it doesn't matter what your academic training, it doesn't matter that one of you is sitting in a U.S. government agency that funds research and is a scientist, and one of you is sitting outside of a U.S. government institution, not a trained scientist, but what you're both saying is the same, and it really just speaks to the importance of bringing all of those different perspectives together. And this is not a scientist-only conversation. This is not an advocate-only conversation. It is really how we pull it all together. And I just can't thank you both enough, because I think one of the things that we always have done in 22 years of HVADs is it is meant, and you did it, Mary, in your presentation, to thank the tens of thousands of trial participants that engage in this work, without whom we'll never find a vaccine, and have done that for many decades, and that is critical. But the other part of HVAD that we do is it really is a chance to re-energize each other and this community and this field of inquiry, and you both have done that so beautifully. So I just want to thank you so much, because we need that, not just on May 18th every year, but always. I also want to highlight, as you'll see on the slide for those still online, there are two webinars next week, one directly related to HVAD and the other not necessarily directly related but clearly connected. And the first one is next Tuesday, and it's work that we, with a number of partners at AVAC, have taken on to try to understand young women and prevention generally, not only about vaccines, but to really try to understand how we can reboot prevention for adolescent girls and young women with a user-centered approach. And we are going to be presenting some updated quantitative findings and some segmentation, because as we all know, putting all young women in one basket as 15 to 24-year-old young women is a ludicrous way to think, and trying to figure out how do we segment them more appropriately to help get prevention to them, whether that is in oral PrEP or with a future injectable, or the dapivirine vaginal ring, or the Adeno 26 vaccine, or a future combination antibody. How do we get them to even think about HIV and prevention? So that's Tuesday's conversation. And then on Thursday at the very same time, 9 a.m. Eastern, and I'll let you all do the math for morning, noon, and night, we have a follow-on to this Vaccine Awareness Day. We touched on it a bit, and a great question, and then an answer from you, Mark, about vaccine hesitancy. Measles was a disease that we thought was largely under complete control, at least here in the United States, and we have seen a resurgence with more new infections this year than since 1994, driven largely by misinformation, anti-vaccine campaigns, and an anti-science approach. And I'm delighted to welcome Heidi Larson, who leads the Vaccine Confidence Project, who also has a long history in HIV, but looking at vaccine confidence generally, and how do we make sure that when we do get an HIV vaccine, because we will, how do we make sure that we can actually deliver it at scale and that people will want it and get it? And she'll talk about that along with Laura Lopez, who is the deputy editor of Efficacy at the South African Mail and Guardian, because so much of what we think about with vaccine education and vaccine hesitancy is how the media engage with this conversation. And Laura and her colleagues at the Mail and Guardian and the Efficacy Health Journalist Center have really been pioneers in helping us communicate and think about how to communicate better about HIV, about health issues generally, and about vaccines. So next week, next Tuesday at 9 a.m. Eastern Time, and then again on Thursday, two different webinars taken together with what you saw today, thanks to Mary and Mark, is a sense of progress, a sense of momentum, a sense of opportunity, and a sense that we have to seize it all together if we ever end this epidemic. So thank you all. Again, lots of materials on the website, and want to thank everyone for sticking all the way through. Most of everyone has stayed on all this time. I want to thank you. More information at AVAC's HVAD webpage, and let's hope this conversation continues well past May 18th, and we get to all work together continually. So thanks again. Have great days.
Thanks, everybody.
Thanks
it was
so
a pleasure.
Thanks so much.
Thank you
Talk
both.
to you
All the
soon.
very best. Cheers.
Okay. Thank you. Bye-bye.